food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomes, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Ramon Velazquez, thank you for joining me.
Thank you for having me here today.
Can you start off by just telling everyone a little bit about who you are and what you study?
Yes, thank you. So I am currently an assistant professor at the School of Life Sciences here at Arizona State University, and I also run a research lab out of the Neurodegenerative Disease Research Center, which is housed in the Biodesign Institute. And our goal within the center is to identify, one, environmental factors that are playing into disorders like Alzheimer's disease and other forms of dementia, as well as developing therapeutic strategies for disorders like Alzheimer's disease. We also study the tauopathies. So the tauopathies include disorders like frontotemporal dementia, where you have a aberrant protein that accumulates known as the tau protein. And we use a variety of model systems in my lab. So here in Arizona, we have a unique program that allows us to actually use brain tissue and other forms of tissue from donors that donate their body to science that have these various diseases. And we're able to acquire this tissue and look at protein dysregulation, validate whether these specific targets that we're studying are actually present in these cases, and also use cell models and animal models to test our hypotheses. My lab has a variety of different students at different stages of their career. So we have undergraduates as well as graduate students that are pursuing PhDs, as well as postdoctoral fellows that finish their PhD and are getting extra training in regards to specific topics they're interested in, creating a path for them to become independent and future leaders in the field.
All right. Well, we're going to talk about a lot of interesting stuff. You've done some interesting work in the realm of neurodegenerative disease, especially environmental factors and chemicals we're exposed to that might play into the pathophysiology of some of these things. I want to have some discussion of neurodegenerative disease to give people a sense for what that is at a high level, and Alzheimer's disease, including what we know about the pathophysiology and how the thinking has changed and some of the controversies in that field over the last few years before getting into some of your recent work. So I want to start very basic. What is a neurodegenerative disease as opposed to, say, some other kind of psychiatric or neurological disorder?
Right. So that's a great question. So a neurodegenerative disease is going to be a disorder where neurons, which are cells in the brain that are critically important in producing our everyday behaviors. They're essentially what allow us to speak, what allows us to listen, to have be able to perceive visual information in our environment. When those cells degenerate because of some sort of pathology or proteinopathy, in this case, a protein that does not belong within those cells, that is what we classify as neurodegenerative disease. These neurons are atrophying, dying in certain cases, and ultimately causing clinical symptoms that begin to compromise the ability of the individual to navigate their environment, and in many cases require a caregiver to take care of them and allow them to essentially survive in the environment.
So proteins start functioning inappropriately, but it's not just that they start functioning in sort of a funny way. They are sick in a way that will eventually kill the actual cell.
Yeah, that is correct. So let's take, for example, Alzheimer's disease, right? Because it's the most common cause leading to dementia. Dementia, as we know, is classified as a decline in intellectual abilities. What you tend to see in Alzheimer's disease is that there's two proteins that are hallmark pathologies, right? And these were actually first identified back by Alois Alzheimer's, a German neurologist who first identified these two pathologies in the brain of a patient known as Augusta de which he was seeing because this patient had symptoms of memory loss as well as sleep disruptions and also hallucinations. In the early 1900s, when this occurred, he was able to actually get the family to agree to look at the brain of Augusta when she passed away, and he identified two proteins that were aberrant and didn't belong in the brain, and this was the amyloid beta plaque and the neurofibrillary tangle. And what we know is that these proteins are a result of changes that occur in within these cells that compromise the function, right? So think about, for example, when we talk about the amyloid pathology, there's a protein known as the amyloid precursor protein that is produced by the amyloid precursor protein gene. And what happens, it's cleaved incorrectly, leading to the production of the amyloid beta plaque, which then begins to compromise the function of neurons communicating. So that's the amyloid beta portion. And then the tau pathology, this other proteinopathy that you see in disorders like Alzheimer's disease and tauopathies, is a result of a protein known as tau that's critically important in neuronal function. But when it begins to go a process that makes it break off its stabilizing components known as microtubules, you tend to see that it aggregates, accumulates, and they can be toxic to the actual cell, ultimately leading to the demise. Now, in addition to those two pathologies in Alzheimer's disease, you also get inflammation, right? So we talk about inflammation. This is essentially molecules that get released by nearby cells known as glia when there's some sort of insult or pathology that you see in the brain. And essentially what you find is that that increase in the inflammation can also harm the neurons themselves because it's chronic, right? Having these inflammatory molecules show up in early phases of these pathologies can be beneficial, but when they're chronically released can actually hurt the neurons themselves. And that ultimately leads to the neurodegeneration.
And so the standard story that I'm familiar with that I think has been the basic model for many years, but I think has some weaknesses in it, although I'm not an expert in this area, is the basic idea here that for whatever reason, things in the cell start to go awry. You get the production of this beta amyloid protein, you get the production of this tau protein. These are things that should not be there, at least not in that form. They start to build up, things get tangled in the cell, and there's just sort of gunk. It gets in the way of normal cellular processes in a way that makes the cell sick and eventually dies. Is that the basic idea here? And how tight is the relationship between those things and the clinical presentation of Alzheimer's dementia?
Right. That's a great, I mean, this is a complex question, right? So I'm going to break it down and answering all these different components. So yes, the idea in general with Alzheimer's disease is that these pathologies build up in these neurons. These neurons are producing the behaviors that allows us to navigate our environment. But when they aggregate and they compromise the communication between these neurons, ultimately your behavior is affected, right? So when you think, for example, of Alzheimer's disease, you can think about Alzheimer's disease as being a disorder that's first identified by the inability to develop new memories, right? Mm-hmm. And that's something that sometimes many individuals are not aware of. They think, oh, I can't remember, who my grandmother is or my grandfather is. That means I have Alzheimer's. Well, not necessarily. The first typical symptom that you see in case of Alzheimer's disease is the ability to actually develop new memories, right? So if we were having this conversation tomorrow and I asked you the question, what did you do yesterday at around this time, and you're not able to answer or remember, that is typically a sign or a feature of the Alzheimer's disease symptomology that occurs early on. And why is that? Because a lot of the pathology that starts, it's typically in that area of the brain where those memories are formed, which is known as the hippocampus, right? So
So
yeah,
people tend to start out having trouble forming new memories, but still retain the long-term memories they've already stored.
correct. And then as the disease progresses, the pathology is found in other areas of the brain, cortical regions. Those are going to be areas more associated with judgment, decision making, long-term memories. Those get compromised. And ultimately, as a disease progression goes through pretty much every area of the brain, you get overall intellectual abilities that decline to the point where an individual requires a caregiver 24/7.
Mm-hmm.
Yeah.
And so if the idea is things like these beta amyloid plaques and or the tau protein build up, and this is really the cause of Alzheimer's, you would expect a very tight relationship. If you go and autopsy the brains of people who had Alzheimer's, you would expect all the people who had it to have these things, and that the worse they had it, the more of these things that they have. Is that the case, or do we find people that have these plaques but don't have Alzheimer's and vice versa?
That's a great question. If you look at a patient that has clinical symptomologies and is diagnosed with probable Alzheimer's disease, the way you confirm this is at autopsy by confirming that they actually had amyloid beta plaques and tau tangles present in their brain. That's how you get a confirmation this person did in fact have Alzheimer's disease, right? So it's at death. But yes, you bring up a good point. There are cases of individuals that have amyloid beta plaques and are clinically fine. They don't develop clinical symptomologies. Why is that? I actually had this conversation this morning with one of my colleagues that runs a program that's really centered around physical activity for individuals that are over the age of 65. And what I was communicating to him is that depending on the cognitive resilience you have, right, which I'll define what that is, that can actually make certain individuals not be affected as much as individuals that have lower amounts of cognitive resilience. So what is cognitive resilience? It's essentially, think about it as use of your brain in capacities that kind of creates a buffer. So these promopathies cannot really affect the cellular mechanisms and ultimately alter the behavior. So engaging in cognitive activities has actually been shown to be one of the mechanisms that can actually lead to protective effects, even in individuals that do show, for example, amyloid beta plaques. So the story is complex, right?
Yeah.
Because once again, if we look at autopsy and here in our local brain bank, we actually see there's some individuals that do develop amyloid and tau pathology, but are clinically okay.
Mm-hmm.
So what we're trying to do with those cases is understand why is that? What changes in those cells do they have that made those neurons resilient, even though the pathologies were actually present?
Mm-hmm. And to the extent that's true, I suppose that would imply that the buildup of these plaques, even in these resilient people that don't present with overt symptoms, that doesn't necessarily mean that the plaques are benign or not doing anything. They simply just might be sort of powering through any extra difficulty, cognitive difficulty these things are causing. It's not necessarily the case that these plaques are totally, just harmless.
Yeah, so there's definitely evidence in the literature if you look at some of the experiments done in cell models, animal models, showing that the amyloid beta plaques can in fact cause some form of toxicity, right? And can elicit inflammatory responses in the brain that can harm the neurons. But once again, the question is, are those given neurons that actually undergo this pathology or have presence of pathology extracellularly? Because the amyloid beta plaques, I just want to clarify, are typically found outside of the neurons, right?
Those are outside.
Whereas the neurofibrillary tau tangles are typically found internally inside the cell, right? So the idea is that it's possible that a neuron, with someone that does have amyloid beta pathology might just have that resilience to resist any sort of interruption that's brought on by the amyloid beta plaque. And these are some of the studies that are currently ongoing. And that's why it's critically important for us to also examine individuals that reach a certain age and are clinically fine to understand why they were resilient and didn't develop aspects of clinical symptomology. Yeah.
Mm-hmm. And I guess there's an issue of cause versus consequence here. Have you guys pinpointed in the field whether these tangles, these plaques, are these a consequence of something further upstream that's going wrong?
Mhm.
or are they the cause of the symptoms
Yeah.
of Alzheimer's?
That's a difficult question. There's a lot of controversy in the field, right? There's one side that believes that the amyloid beta plaques are causative of the clinical symptomology because what they highlight is that the amyloid beta plaques are usually the first proteinopathy that shows up in Alzheimer's disease. But then as you progress and down the different pathologies, you then typically get the tau pathology, right? And it's not really until the tau pathology is present that you really find signs of clinical symptomology. What
Mm.
you also find there are synapses are lost, right? So synapses are, you can think about a space between two nearby neurons. They communicate chemically that way, right? And by communicating and strengthening those synapses, you're able to produce memories. You're able to engage in various behaviors. And so when those synapses are lost or don't are not properly functioning, those behaviors are essentially compromised. So if we talk about the trajectory, it is shown that amyloid typically does occur first, and then you typically have the synaptic loss and then the tau pathology. But it's not typically until the tau pathology is present where individuals do start showing signs of clinical symptoms. Yeah.
I see. I see. So the basic order of operations here seems to be that these these beta amyloid plaques outside the cells start to appear. At that
Mhm.
point, you may or may not have any symptoms that are detectable. Oftentimes, you won't even have symptoms, especially in more resilient people. Later on, it tends to be that you get these tau protein tangles building up inside of cells. That's typically when you start to see the clinical manifestations of the cognitive issues that people have. So, sort of putting that all together, I've seen people sort of point to autopsy data and say, like, well, we have plenty of brains where they had all these beta amyloid plaques, but they didn't have Alzheimer's disease. It sounds like what you're saying is basically you were probably seeing them at a relatively early stage, and or they may have been a more resilient person, and they could very well have developed symptoms later on if they had survived longer.
Correct, because it also depends on how old that individual was, how, if they were, if we're talking about someone that's 55 years of age, maybe they're still early phases, and they might actually be in, what we call preclinical AD. It's not identified. There's no symptomology. They haven't even reached the level of mild cognitive impairment at that point, right? If they were, but we won't know that because there's no way to know that because the individual has died at that point, right? But I
Yeah.
do want to make one point because I think what this points to is that the amyloid, in order to have a diagnosis of Alzheimer's disease, you need to have the presence of amyloid and tau pathology, right?
Mm-hmm.
But what we find is that typically the clinical symptomology comes closer to the tau pathology.
Mm-hmm.
Other side of that solidifies that the tau pathology really might be more of a driver of those clinical symptomologies is that you have tauopathies, disorders where you don't have amyloid beta plaques, just tau pathology, and those individuals also have clinical symptomology. So in frontotemporal dementia, for example, you find the tau pathology, chronic traumatic encephalopathy, which is a result of traumatic brain injuries. You tend to find the tau pathology that leads to these clinical symptoms. And Pick's disease is another example. So highlighting that the tau pathology might be more indicative of the actual decline in clinical
Mm-hmm.
symptomology.
Mm-hmm.
Yeah.
And I just want to get really concrete about this tau pathology. When we say tangles in the cell, is that just tau protein aggregating together? Is it microtubules getting twisted together? What is the tangle?
So let me answer that question by describing what tau is and what happens in this process. So the tau protein is, you can think about this within neurons as a stabilizer of railroad tracks, right? So microtubules are essentially these various proteins that are critically important in creating the architecture of the neuron, but also are important in allowing proteins to move from one part of the cell to the other, right?
Mm-hmm.
So the neuron has various different compartments, and you need to traffic proteins from one part of the cell to the other to allow the neuron to behave in a certain way and produce given behaviors. And what we find with the tau pathology is that, well, first of all, the tau protein stabilizes those microtubules. So imagine a railroad system and railroad tracks with stabilizers so those railroad tracks don't, when the train is riding on it, the train doesn't fall off the actual
Mm.
track. The tau protein stabilizes that. However, in Alzheimer's disease and other forms of tauopathies, there's a process where that tau protein undergoes what's called phosphorylation, and specifically hyperphosphorylation. It's known as a post-translational modification where the protein tau, there's addition of phosphate groups that cause it to be removed from the microtubules. But in Alzheimer's disease, it happens in a very fast, accelerated manner to the point where the tau can no longer stabilize the
Yeah.
microtubule. So the microtubules disintegrate. They can no longer allow proteins to traffic from one part of the cell to the other. And then that tau protein that's now phosphorylated, hyperphosphorylated, then begins to aggregate ultimately into the neurofibrillary tangle. So you have two problems within that neuron. One, the microtubules are no longer stable. You have axonal transport issues, so you can't move proteins from one part of the cell to the other, which means the cell cannot function properly. And two, you have the aggregation of this toxic form of tau, the
Mhm.
neurofibrillary tangle within the actual cell.
I see. So in all cells, you've got microtubules, you've got a cytoskeleton, literally a molecular skeleton that gives the cell its rigidity and its geometry and its form. But also, these are important for trafficking purposes. So you use the analogy of a railway. You can think of the microtubules as being like railroad tracks because proteins literally get shuttled from, say, the nucleus out to some other part of the cell or back and forth or whatever. And this happens in all cells, but this is probably especially important in some sense in neurons because they have this odd shape where they can be very long. And you've got to traffic stuff, say, all the way to the synapse. And I would
Right.
imagine that the reason that some type of pathology like this, a disintegration of the railroad tracks and these tangles based on this hyperphosphorylated tau protein, that manifests in cognitive issues like memory issues, because one of the ways that memories form, an important process, is the shuttling of proteins back and forth to the synapses so that they can be strengthened or weakened or whatever. And so naturally, if you get these tangles and the microtubules can't the tracks can't be laid down right, then you're not going to be able to engage in the types of synaptic plasticity that our cognition relies on.
Yeah, that's 100% correct. Thank you for summarizing that. And you can also think about it as the synapses and ultimately the what we call the postsynaptic cell, right? That is the neuron on the receiving end. So you have a presynaptic cell and a postsynaptic cell. The postsynaptic cell has the ability to upregulate the number of receptors that the presynaptic cell releases the what's called a ligand and binds to those receptors to activate it, right? It relies on axonal transport to do that, to build those proteins that are
Mhm.
necessary to increase what are called dendritic spines. And when the axons are no longer able to do that because microtubules have disintegrated and the proteins can not move from one part cell to the other, then you ultimately get compromisation of synaptic plasticity and the ultimately the inability to reinforce certain behaviors such as developing learning, developing memory. Yeah.
Mhm. And so if we just sort of move up the causal chain here, you said it was hyperphosphorylated tau. So the tau protein is normally there to stabilize. It's supposed to be there. It's to stabilize these microtubules and enable all of that trafficking and stuff to happen. It becomes hyperphosphorylated in some cases. And this
Mhm.
is what sort of initiates the problems we were just talking about. What is the origin of the hyperphosphorylation of tau protein? Do we know that?
Yes, we do know that. So phosphorylation of tau proteins and various other proteins occurs through kinases, right? These enzymes that add the phosphate groups. What we have found, which ties it back to actually the amyloid beta hypothesis, is that the amyloid beta triggers a lot of these kinases to upregulate that then are directly phosphorylating the actual tau protein. So you get more amyloid beta, an increase in certain kinases and here's some examples. One of them is known as GSK3 beta. The other one is CDK5. One that we study in my lab is known as DYRK1A. The activity of those kinases goes up, which means that the protein itself is more prone to being phosphorylated. And that is why you get the hyperphosphorylation that actually occurs.
Mhm. And are those kinases specific for tau protein, or do they just kind of phosphorylate a bunch of stuff?
They can phosphorylate other proteins as well, but they tend to adhere or have a predisposition to the tau protein. Yes.
Mhm. And so I have not followed it super closely, but I've followed it a little bit over the last I don't know, several years, some number of years, there have been some controversies and some high profile papers that were very instrumental in bringing this sort of hypothesis to where it's at today that were retracted and there seems to be some controversy there. What exactly is going on there, and how has this general theory been impacted by that?
Yeah. Well, so there have been a lot of, in the last couple of years, and I do want to say this is a result, a lot of the development of AI, being able to screen manuscripts and see if there's some alterations in images, which unfortunately are showing up. There have been publications that have been pulled and retracted because the data is not reliable, right? And some of them have been tied to the amyloid hypothesis, right? But the evidence the evidence that points that amyloid beta is definitely present in Alzheimer's disease is still there. There's many papers that support that, right?
Mm-hmm.
So a lot of the papers that have been retracted in the last couple of years that are tied to that are tied to various, forms of amyloid beta, et cetera. But the general premise tied to the amyloid hypothesis that it is present in Alzheimer's disease and it could actually initiate a lot of these kinases or activate them that lead to tau is still sound. So the amyloid hypothesis still has a lot of evidence to back it. Yeah.
Mm-hmm. And if I'm understanding you correctly, sort of my read of what you've said in this whole thing is that beta amyloid plaques tend to precede the tauopathy that seems to be particularly important to Alzheimer's. But what causes the tauopathy is increased activity of these kinases that hyperphosphorylate the tau protein. And one possible initiating factor of that kinase activity going up is amyloid beta plaques. But I would imagine logically speaking, that's probably not the only way that can happen. There might be alternative ways that can happen.
Right. So there's also evidence suggesting that inflammation in the brain can initiate these kinases, right? So going back to the primary tauopathies, frontotemporal dementia, talking about chronic traumatic encephalopathy, you usually see tau proteins that build up there, and there's really no evidence of amyloid beta being present. So yes, the question is, why is that? And what we're finding is that for example, the example of CT is a good one, chronic traumatic encephalopathy, that's typically triggered by repeated head blows, right? Traumatic brain injuries. And we know with those injuries, you typically have inflammation that is elevated in the brain and chronically elevated. So there is evidence actually now showing that having increases in inflammatory molecules in the brain can actually lead to increases in those kinases as well that can then initiate the hyperphosphorylation and those neurofibrillary tau tangles. Yes.
I see. So if we think in like a 2x2 grid here, tau protein present, absent, beta amyloid plaques present, absent. It sounds like in almost all cases of Alzheimer's, when you do the autopsy, you're going to find both tau and plaques, but perhaps in a minority of cases, you find the hyperphosphorylated tau and the tangles without plaques because it was caused by some other thing. And you're essentially never going to find, you might find, an absence of the plaques, but you're never going to find pathology in the absence of tau. And you're usually not going to find it in the absence of plaques, but sometimes you will.
Yeah, I mean, I think that's a nice summary, and that really, highlights the complexity of these diseases, right? That some of them are initiated by an aberrant, protein like amyloid beta, and some of them are by the inflammatory molecules because of repeated head traumas and that sort of thing. I think the point here that I want to make is that it goes back to the general premise that ultimately the tau tangles is what you really see that drives a lot of these different clinical symptomologies, right? But the evidence that's being put out there now tied to the therapies that actually target amyloid beta, right? Like lecanemab, that's now clinically approved. Their hypothesis is that if we halt the amyloid pathology, that can lead to a halting of the actual tau pathology, right? And there's actually some evidence of that. If you look at a presentation that was given by Biogen in 2024 at the Alzheimer's conference, they actually show that individuals that have been on lecanemab now, this antibody therapy that essentially tags the amyloid beta plaque for degradation, they actually also show some signs of decreases in tau pathology as well, right? In these Alzheimer's cases. Yeah. So I think that points back to the fact that the amyloid beta plaques likely are initiating a cascade of events tied to the tau pathology, whereas you can also get the tau pathology due to other insults like the traumatic brain injuries. Yeah.
Got it. Got it. So what seems to be really key here is something, which can often be the beta amyloid plaques, but it could be other things. But something is going to trigger kinases to become more active and hyperphosphorylate the tau protein. That's when you really start to have a problem. Thinking ahead to some of the other stuff that I know that you work on that we're going to talk about, you mentioned traumatic brain injury. You could just get, blunt force trauma, which triggers inflammation that could start to initiate this hyperphosphorylation process independent of, say, beta amyloid plaques. I would imagine that in theory, at least, there could be, say, chemicals or toxins in the environment that might upregulate kinase activity and lead to the same result. What do we know about possible environmental toxins that might tie into Alzheimer's disease?
Yeah, there's a lot of evidence coming out now, looking at various plastics and how plastics that might actually migrate into the gut microbiome. There's a lot of connection between the gut microbiome and the brain. Might actually show up in the brain. These plastics might show up, and we know that anything foreign that has the ability to cross the blood-brain barrier, right? Blood-brain barrier, you can think about it as vasculature that has a variety of barriers preventing things from getting into the brain. Because what you want to do is you want selective things to get into the brain, chemicals, compounds into the brain, because if they're pathogenic, for example, or they're viruses or pathogens that can actually harm the neuron, and if the neuron is damaged permanently, in many cases, they don't regenerate, right? So what you're finding is that plastics, for example, have the ability to get through this barrier. And there's actually recent papers that are actually showing that it's present in the brain, which is very alarming. In terms of other toxins, we've studied quite a bit the herbicide known as glyphosate, which is a product that is used, a chemical that is used to control unwanted weeds in agricultural communities, right? And the idea with glyphosate is that it's deemed safe to mammalian species because the way it works is by blocking a pathway known as the shikimate pathway. Shikimate pathway is found in plants, and essentially, if you block that pathway, you block the production of important proteins that leads the plant to die, right? But the idea when it was first initiated was that, or when it was first used in the 1970s, was that mammalian species do not have a shikimate pathway, so any glyphosate that you consume is literally going to just be excreted out of your body. But we're finding that that's not the case. As you've probably seen, there's been a lot of research and litigation tied to a lot of effects that glyphosate can have on cancers, right? So a lot of non-Hodgkin lymphomas, a lot of blood cancers, et cetera. Our goal with the research we just recently published in the last couple of years was to understand what are the effects it can have in the brain. And what we're finding is pretty daunting, to say the least.
Yeah. So before we get into your work, I want to build some concepts for people here. So we'll sort of apply some scientific thinking to a topic that sort of bleeds into other issues. So glyphosate, this is an herbicide, as you said, basically a weed killer. This is the Monsanto Roundup product or whatever it's called. It's used all over the place. It's a very, very highly used product. So glyphosate is in the environment to a large degree. I think most people know that at this point. The idea when this thing came out was, okay, well, in order to develop this, they probably figured out, we want a chemical that's going to disrupt some pathway in weeds and plants that's not found in animals and humans. Therefore, it's only going to affect the plants and it's not going to do harm to us. Another way of saying that is, if we develop a compound that disrupts a pathway in plants that's not found in animals, and we make the critical assumption that it's not also impacting other pathways in some way, shape, or form in the body, then it would be safe. And I would imagine that the lawyers at these companies probably used arguments of that kind to get it deemed legally safe, or safe in a legal sense. But, just thinking sort of, a priori here, what are the odds that a compound like this, even though it operates for this particular application on a plant pathway that's not an animal, what are the odds that it wouldn't have any other effect in an animal body?
Yeah, that's a great question. I would say that based on what we know, based on the literature, I'd say the odds are it probably is affecting, it could affect other specific pathways. And one of them in which we're seeing it's affecting is that it's initiating various aspects of inflammation, right? So inflammation, once again, is tied to many disorders, right? Many diseases, not just, brain diseases, but also peripheral body diseases. And what we're finding is that exposure to this herbicide, glyphosate, is in fact inducing inflammation in the periphery body as well as in the brain. But what's really fascinating as well is that yes, the assumption was made that we couldn't process or metabolize glyphosate, but a paper came out in 2020 that actually showed humans can and mammalian species can.
Mhm.
And how is that? The gut microbiome has bacteria. That bacteria has the ability to metabolize glyphosate into what's called amino methylphosphonic acid, which is AMPA. And studies have shown that AMPA itself is pretty toxic to the body.
Hmm.
So what we're ultimately finding is that glyphosate does in fact have the ability to affect other signaling pathways in detrimental ways. Yeah.
Hmm. So let's talk a little bit more about the chemistry of glyphosate. What kind of molecule is this? Is it big? Is it small? Does it resemble anything that's naturally found in the body?
Yeah, it's a small molecule. In terms of its resemblance to anything else in the body, nothing comes to mind. But it's a small molecule that can leak through the blood-brain barrier pretty easy.
Hmm.
And we've seen that it has the ability to do this using cell models as well as in vivo models. Yeah,
Mhm.
in terms of its homology to other endogenous molecules in humans and in mammalian species, nothing comes to mind. Yeah.
Mhm. And so when it was originally deemed safe, I would suppose this is probably back in the 1970s there or thereabouts, when it was deemed safe because it operates, its primary mechanism for its application is through a plant pathway that we don't have. It sounds like no, I mean, no, did no one just check to see if it can be metabolized by the human or get through the blood-brain barrier? Or why did the work that you just cited, why is that so many years in between that work getting published and the introduction of this thing into our environment?
Yeah, I think the answer to that is the way that I would answer that question is that, some of this work was investigated. They would give glyphosate to rodents, for example, and looked at toxicity effects, and they didn't
Mm.
really find a lot of toxicity.
Who's they?
Researchers, scientists, there is evidence. The EPA did some of this as well, and scientists by the EPA, and didn't really identify a lot of toxic effects, right? But they also did different dosages, right? And we do actually have what's called a NOEL, a no aversible adverse effect level that is published actually by the EPA and other organizations as well. At a certain amount or a certain dose, there is actually toxicity that is present, right? But in terms of, yeah, examining whether this actually blocked a shikimate pathway, the answer always was, well, there is no shikimate pathway in mammalian species, so it's not going to be present there. So what do you really look at in that regards, right?
Yeah, yeah.
Yeah.
And I would assume, I'm sort of, I guess, foreshadowing a little bit. I would assume that a critical assumption that goes into that type of work is, well, there's a difference between acute and chronic toxicity, and there
Right.
could be a difference between something being toxic, at a given dose right now that you inject into an animal versus, say, toxic if something accumulates in the body over time. And if the only way to determine if that's true is to do very long-term studies, you might not pick that up in the types of studies that would be done initially.
Absolutely correct. And the thing we have to keep in mind is, in addition, is that these studies are going to be done in rats. They're going to be done
Yeah.
in animals, right? You can't dose a human. That's not allowed. So you do these studies, and even if you did the longest dose, time frame possible, rats and mice don't live, the same time frame that a human does, right? So you're probably talking about rodents 28 months at max. So even if you dose an animal that long, it's still not going to be the same as if you dose a human for 75, 80 years to look at the impact, right? But I will say that some of the work that's coming out that is starting to shed light in this, and this has come out in the last few years, which also made us very interesting, is that if you look at the circulating levels in urine of humans in small sample studies, you find that about 90% of us have glyphosate circulating through our bodies at all times, right?
At
That's
some level.
At some level, correct. Now, what those levels are, they're going to vary. We are actually currently looking at what the differences are, for example, between rural versus urban communities, right?
Yeah.
You would expect in rural communities where there's more agricultural fields and more of this product spray that there's probably going to be higher amounts of glyphosate present, right? But the other component is that, a lot of the foods that are consumed on a daily basis by consumers do have glyphosate present in them as well, coffee and among other items as well. Yeah.
Yeah. So this isn't just found in like a Roundup bottle that you can buy and use in your home backyard. This is used at an industrial scale.
Correct. Yes, it is. So just to give you an idea of the amounts and how widely used glyphosate is, we're talking about 300 million pounds in the United States per year
Mhm.
at the moment. Yeah. So it's used very heavily to control unwanted weeds in these agriculture communities, which ultimately, are growing all the product and produce that we as consumers purchase so we can, eat and survive. But that means that, we're being exposed to all these to this specific herbicide, yeah, because of that. Yeah.
Um, Well, there's a number of ways we can go with this. Let's talk next about what you guys are finding in your lab. So you've done studies where you actually give glyphosate to rodents for
Mhm.
quite a long time, especially relative to their lifespan. You give it to them, you look to see what happens. You also give it to them, and then you stop and you wait to mimic, say, an exposure that might happen for a number of years early in life. And then, someone goes away. Are there long-lasting effects? What are some of the studies that you've done, and what was the design?
Yeah, so the first study that we really wanted to conduct was back in 2020, really after the groundbreaking work showing that glyphosate can actually be metabolized by mammalian species, and tied to the gut microbiome, metabolism, et cetera. So what we wanted to do is to simply say, if we introduce mice to glyphosate, these are normal mice, not genetically altered mice, and I'll talk about what those are in the next couple of minutes.
Full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.